Thank you. So hello, everyone. Like she said, my name is Brianna Edwards, and I'm a PhD student from the Department of Mathematical Sciences at Florida Institute of Technology, and I'll be presenting this work titled Multisample PCA Applications for Reconstructing Structure of Biological Tissue, and this was done in conjunction with my PhD advisor, Dr. Vladislav Bushtinov. So I'll start out by discussing some of the motivations behind this research, and then we'll get into some of the mathematical formulation. And finally, I'll present our first results and their implications. So EIT is a really cool, relatively novel medical imaging technique where electrodes are placed on the skin around a boundary of a particular region, such as the breast, the lungs, or even the brain. So in this particular scheme that we use in our research, voltages are applied at these electrodes and the resultant currents are recorded. Other schemes do the opposite. So the current is injected and then the resultant voltages are used as the measurements. So as we know, electrical current flows along the path of least resistance, and cancerous tissue has a higher conductivity than normal tissue. So these measurements can be used to detect abnormalities in the body. The main benefit of this method is that it's completely non-invasive, so no contrast agents are needed, the patient doesn't feel a thing, and they're not subjected to any harmful radiation. It's also more cost-effective, especially because it requires very little hardware, as you can see in this image here. It's just a few electrodes and wires. So there's a few challenges with this. The main challenge is that for getting high resolution images, this problem is very ill posed due to the fact that very small number of measurements are available. And this is just because you can only fit so many electrodes on a particular area of the body. Also, signal loss and interferences come into play when too much hardware is present in a given area. And if the electrodes aren't properly attached to the skin, then that can also distort some of those measurements. Lastly, the solution is non-unique because we have a 3D area being projected into a 2D space where the images are obtained on your computer. So now for some math. So EIT is based on the electrical charge conduction problem. This is our spatial domain, our omega, and as you can see, this is just the area inside of a circle or the volume inside of a cylinder, depending on if we're talking 2D or 3D. Sigma is our electrical conductivity, and that is our control variable, where U is our unknown state variable that can be found by solving this PDE, which is the forward problem. So this second line here describes the portion of the boundary that's not covered by the electrodes, and you can see the potential flow or the current into that part of the body is 0. And then this part describes the portion of the boundary that is covered by the electrodes, and this is dictated by good old Ohm's law. So Z is our surface impedance or our electrode contact impedance with the skin, and big U is our applied voltages at those electrodes. And the sum of these electrodes potentials here is 0, and that's just due to the ground condition. Okay, so from this we get two types of current. We get this I star, which is the current that we can measure at those electrodes, and then we get this current that we get from solving the forward problem using the potential, but that's provided that our sigma is known. So this is when it gets kind of fun. The goal is to minimize this discrepancy between the calculated current and the measured current to get our most accurate representation of our sigma. And we use this gradient descent algorithm to perform this optimization, and the gradient is performed using adjoint-based techniques. This tau here is our step size in the gradient descent algorithm, and we use a line minimization search to get an optimal value that maximizes the performance of this optimization. Now, to perform actual computations, our sigma, which is a smooth function in our functional space, needs to be represented in a finite dimensional parametrized version of this solution space. So this is computed over multiple iterations. Also in this space, sigma is highly irregular, so it also needs to be regularized in some way. So we do this by creating an augmented cost functional where we add this regularization term that is weighted by this coefficient lambda. So CR here, which is a matrix that describes the connectivity between different regions of our discretized solution space. So say you have element 1 of our space and element 300. If they're really close together, then that number at the space in the matrix 1, 300 is going to be close to 1 if they're close together. And if they're really far apart, it's going to be closer to 0. And this is just a covariance matrix based on these sample realizations that we have created, which I'll get more into in a little bit. So now we use principal component analysis, also known as proper orthogonal decomposition, for 3 purposes. So first, we want to change out our highly correlated sigmas to uncorrelated xi's. So, and then also we want to reduce the dimensionality of our solution space and to use this prior information that is in these sample images that I mentioned before. So our old controls sigmas and our new controls xi's are related using this top left equation here. And phi is our PCA transformation matrix. And that's created using this generated set of samples, which I'll give examples of in a bit. This big sigma here is a diagonal matrix of all the eigenvalues ordered from greatest to least. And then this U here is the eigenvectors in that corresponding order. So this phi matrix is now truncated, or our representation of our psi space is truncated. And there's different ways that you can decide how to truncate this matrix and how much of that energy or information you need from that matrix. So usually phi is created using one set of sample images. But however, this research focuses on the integration of multiple sample sets into to create our phi. And this is called multiple factorial analysis. And we use this for our PCA. So we can create different sample sets, each with a different defining topology. And the goal is to create an automatic procedure for combining these multiple factors in an ideal way. So we scale each of these new resultant sigmas by these alphas, and this is these all sum to 1 to make a convex combination. And we can also choose the different number of components that we use from those phi matrices. So how we want to truncate each of those matrices individually. So both of these variables now we put into our optimization problem. So now we're optimizing over xi, which is the reduced dimension uncorrelated variables that represent sigma, alpha, the weights of each factor, and n xi, or the number of components used from each sample set. So say we have 16 electrodes in our 2D model placed around a boundary of a breast. So we want to see if there's any malignant spots inside of this boundary. And we know that healthy tissue has a conductivity of about 0.2, and cancer-affected tissue has a conductivity of around 0.4. So you can see it represented in this image. This is the cancerous spot, this is the healthy tissue, and this is what it would look like in a potential model. So we break up our space like this, and then we create our synthetic measurements that I explained in the beginning. And this would be a representation of how the current flows through this two-dimensional cross-section based on given potentials that are applied at these electrodes. So we've created 5 models for testing our algorithm on. This one here, model 1, is various size circles placed randomly. Model 2 is same size circles placed in a symmetric fashion. Model 3 is slightly elliptical ellipses placed also in a symmetric fashion. Model 4 is non-symmetric and more highly elliptical ellipses. And model 5 is perhaps the most useful. This was created using filtering techniques on an actual MRI of a true breast cancer patient. So here we show our sample sets, or how we create our sample sets, or our sample sets of images that we use in our PCA. So we have 6 sets, A through F, and they're each related by the size of the circles used. And the amount and configuration of these circles is random from image to image. There's 1,000 samples in each of them, and there's 1 to 10 circles in each image. These, like I said, there's 1,000 samples in each, so this is just a random assortment from some of the samples to show how these might look. So our preliminary research was an experiment where we reconstructed these 5 models that I spoke about using each of these sample sets individually to see how they compare with each other. And we project each of the models from the original sigma space to a reduced dimensional representation of our size space using these different sample sets from A to F, and a different number of principal components each time, each run through. And this is estimated for simplicity with accumulated variance rather than number of components. So model 2 was quite interesting because the sample sets that showed the better fit within the energy range of around 80 to 90% were the sample sets that had similarly sized circles to those shown in model 2. And that's namely samples C and D. So on the left is the accumulated variance versus the objective function, and on the right is the accumulated variance versus the L2 norm of sigma, or the error, the total summed error compared to the true model image. 80 to 90% is a good range to look at because we have a good chunk of that variance or PCA energy going into this process, but it's not up in the 100% range. It wastes a lot of computational cost, a lot of time. So that's a good range to look at there. So here we have model 4 on the left, and we assume that these long lengths of these ellipses corresponds to models D and E, and the short width is closer to samples B and C. So this sort of, again, a sort of resonance can be seen where D and E shows steady convergence or performance changes over number of principal components, and the samples with the smaller circles have a slow start at first, but then they get faster convergence as you get into this 75 to 80% range. Model 5, also very interesting because sample F, which is the smallest circles, shows pretty poor performance when compared to those larger circle sizes. And this told us that we need to find some way to work out these jagged edges and irregular shapes that you see in true cancer images. So this led us to believe that perhaps we could combine these sample sets through multiple factorial analysis to get some better results. So this is the bulk of the results here. You see 2 graphs. The one on the left shows the objective function for each model using 3 different optimization processes. So these green dots here, they show the optimization using only Xi. So this is the original that I explained, except the only difference is we're using multiple factorial analysis with the number of components used from each sample and the weights of each sample approximately equal and fixed. And then these blue dots are where we're optimizing over Xi and alpha, or the weights of each of those sample groups. And then the red dots are optimizing over Xi and nXi, or the number of components used in that PCA transformation. So clearly the objective function is significantly improved between the red and the green dots, and depending on the model, the blue dots show some improvement as well. This could be explained by the fact that having the weights alpha as controls may be kind of considered similar to running the classical optimization, because these weights are present inside the Xi controls as constant multipliers. At the same time, these error norms that you see in the right figure, adding weights alpha as controls shows that we're converging to a solution that is not the same as the classical approach per se. So on the other hand, when we change the number of principal components, so the red dots, we see that we get a solution in the neighborhood of the classical approach, but with significantly improved measurement match as demonstrated over here on the left. So these results are pretty promising because they show that they're consistent between models, and it shows that they're very sensitive to these changes in alphas, and n psis. We have no doubts that the results will be further improved by changing various parameters in our computations, including experimenting with a finer mesh size during the discretization process. The biggest challenge is figuring out how to make all these control variables active in the same process. So psi, alpha, and n psi. So overall, we've shown that these areas are reconstructed best when the sample groups show a similar topology to the areas that we're trying to reconstruct. MFA has the potential to improve these qualities of these images. And in the future, we aim to, like I said, experiment with all three controls and try sample groups that contain shapes other than circles. The ultimate goal is to extend this to a 3D configuration, which is much more true to real life situations as our bodies are 3D and would address the problem of electrical current that comes out of the 2D cross-sectional area that we're looking at, which would increase the reliability and accuracy. So that is all, and thank you, and I will take any questions.